I recently came across a paper that I wanted to share with you and get your thoughts on. Part of me was incredibly impressed by what the researchers managed to build and by what this small piece of living neural tissue was capable of doing. But another part of me felt slightly uneasy about where technology like this could eventually lead. And beyond the engineering, the experiment raised some deeper questions for me about neural tissue, computation, and what exactly separates a living brain from a biological machine. But before we get into any of that, let's walk through exactly what the researchers did. Today, we're looking at a paper published in Nature Electronics in December of 2023 titled Brain Organoid Reservoir Computing for Artificial Intelligence. Before we get into what the researchers actually built, it's worth understanding the problem they were trying to solve. Artificial intelligence has advanced rapidly through the use of artificial neural networks running on silicon computer chips. But despite their success, these systems can be slow and extremely energy intensive to train. One reason is that traditional computers physically separate memory from processing. Information has to move back and forth between where it is stored and where it is used, creating what is known as the von Neumann bottleneck. Researchers have spent years trying to design hardware that works more like the brain. Engineers have developed neuromorphic chips, including devices known as memristors, that can retain traces of previous electrical activity and reproduce some brain-like forms of memory and information processing. But these electronic systems still capture only part of what some biological neural networks can do. In the brain, memory and processing occur within the same living network. That network can reorganize through synaptic plasticity and learn from incomplete or noisy information without requiring the same level of precision as conventional computing systems. The energy difference is also striking. A human brain operates on roughly 20 watts, while the authors cite an estimate of about 8 million watts for current hardware running a comparable artificial neural network. So the researchers behind this paper asked a much more radical question. Instead of continuing to build electronics that imitate the brain, what if living brain tissue became part of the computer itself? And that is exactly what these researchers set out to test. They called their system Brainoware. At its heart, it was a small human brain organoid grown from pluripotent stem cells. This wasn't a complete brain. It was a three-dimensional cluster of developing neural tissue containing neurons, astrocytes, neural progenitor cells, and other cell types that naturally formed a living network capable of producing electrical activity. The researchers then placed that organoid onto a high-density multielectrode array. Think of this electrode array as the organoid's interface with the outside world. Some electrodes deliver carefully designed electrical stimulation into the tissue. Other electrodes recorded how the living neural network responded. At this point, it's important to understand that the organoid wasn't operating by itself. The entire system was a hybrid. A conventional computer first converted information into patterns of electrical stimulation. Those electrical signals were then delivered into the organoid. Inside the organoid, the living neural network transformed those inputs through its own electrical activity. Finally, another computer program analyzed the organoid's responses to determine the final output. The researchers described the organoid as an adaptive living reservoir. In other words, instead of trying to program every computation directly, they allowed the living neural network itself to transform the incoming information. Unlike conventional reservoir computing, this reservoir wasn't fixed. As the organoid received repeated patterns of stimulation, its neural connections could reorganize through synaptic plasticity. That meant the reservoir itself could change with experience. And according to the authors, that ability to continually reshape its own network is what allowed Brainoware to improve over time. The next question then was whether the living reservoir actually possessed the properties needed to function as a useful computing system. To answer that, the researchers weren't interested in whether it could recognize speech or solve mathematical problems just yet. First, they needed to know whether the organoid behaved like a computational reservoir at all. So they delivered carefully controlled electrical pulses into the organoid while recording its electrical activity through the multi-electrode array. And then they looked for several key properties expected of reservoir computing systems. First, they tested whether the organoid responded non-linearly. In other words, if they doubled the strength of the electrical stimulation, would the response simply double? It didn't. Instead, the organoid transformed those inputs into a much more complex way, one of the fundamental properties required for useful computation. Next, they look for something called fading memory. After each electrical pulse, the activity didn't immediately disappear. Instead, it lingered briefly before gradually fading away. And that means the network retained information about its recent past, an important property for processing sequences like speech. They also found that when electrical pulses arrived close together, the responses accumulated rather than resetting after every stimulation. Finally, they tested whether the organoid could distinguish different patterns of stimulation across the electrode array. It could. Different spatial patterns produced different patterns of neural activity, showing that the organoid wasn't simply reacting to electricity. It was processing spatial information. Taken together, these experiments show that the organoid possessed the core properties expected of a computational reservoir. The stage was now set for the real test. Could this tiny piece of living brain tissue actually perform useful computational tasks? To find out, they chose speech recognition. The researchers used 240 recordings of the Japanese vowel sounds A and E, spoken by 8 different men. Those audio recordings were converted into patterns of electrical stimulation and delivered to the organoid through the multi-electrode array. The organoid responded with its own patterns of neural activity. A conventional computer then used a logistics regression model to analyze those responses and classify which speaker had produced each sound. Before repeated training, Brainoware identified the correct speaker with an accuracy of about 51%. The researchers then presented the same organoid with 4 training epochs over 2 days, one every 12 hours. After training, its accuracy increased to about 78%. But here's what I find even more fascinating. The organoid itself appeared to be changing. When the researchers compared its functional connectivity before and during training, they found substantially more changes during training. Some connections strengthened, some weakened, new connections appeared, and others were pruned. In other words, repeated stimulation was not merely passing through a fixed network. The living neural network was reorganizing as it encountered the data. The authors suggest that this biological plasticity helped the larger Brainoware system improve its performance over time. But speech recognition wasn't the only task they tested. The researchers wanted to know whether Brainoware could tackle a completely different kind of problem. Instead of recognizing sounds, they challenged it to predict the behavior of a chaotic mathematical system known as a Hénon map. You might not know what a Hénon map is, and honestly, neither do I, but the important point is that this is a difficult prediction problem in which tiny differences can quickly grow into very different outcomes. The researchers converted the mathematical data into patterns of electrical stimulation and delivered those signals into the organoid, just as they had done with the speech recordings. Once again, they compared Brainoware's performance before and after training. And once again, training made a dramatic difference. The regression score increased from about 0.36 before training to about 0.81 after 4 training epochs. But then the researchers performed what I think is the most important experiment in the paper. They repeated the training after blocking activity-dependent synaptic plasticity using a compound called K252A. This time the score improved only slightly, from about 0.31 to about 0.39. In other words, when the organoid could no longer recognize its own connections normally, Brainoware largely stopped getting better at the task. The researchers also tested the system without an organoid present. In that condition, the regression fell to 0. Together, these controls strongly suggest that the improvement was not simply coming from the external computer or the electrode array. The adaptive properties of the living neural tissue itself were playing an essential role. So, what did the researchers conclude from all of this? Their central claim was that the human brain organoids could serve as a new kind of reservoir computing hardware. The organoids were able to self-organize into functional neural networks, respond to electrical stimulation, briefly retain information, distinguish spatial patterns, and reorganize their connections during training. That gave Brainoware several of the properties the researchers were looking for in an adaptive computing system. They also show that the system could be used for two very different tasks: speech recognition and prediction of a nonlinear chaotic equation. From the author's perspective, this suggests that the living neural tissue could eventually inspire more adaptive forms of artificial intelligence while potentially reducing some of the energy use, training time, and heat production associated with conventional hardware. But they were also clear that this was an early proof of concept. Brain organoids are hard to standardize and difficult to produce consistently. Some can suffer from poor oxygen supply, dead tissue at the center, or limited viability. The flat electrode array could only communicate with a limited number of neurons near the organoid surface, so there was limited access. The current system also required supporting hardware like an incubator, a conventional computer, and other equipment, so the low energy use of the organoid itself does not yet mean the complete system is highly energy efficient. And the methods used to encode information into the electrical stimulation, record the response, and decode the output still need substantial improvement. So the researchers were not claiming that living computers are ready to replace silicon. They were showing something more preliminary, but still remarkable. Living human neural tissue could be integrated into a hybrid computing system, adapt their biological plasticity, and contribute meaningfully to computational performance. So that's the experiment, and honestly, I'm not sure what I think still. On the one hand, I'm incredibly impressed. The researchers didn't just grow a brain organoid, they integrated living human neural tissue into a computer system and showed that it could adapt, reorganize itself, and contribute to solving real computational problems. That's remarkable. But if I'm honest, it's also a little unsettling. One thing I kept thinking while reading this paper was how similar the overall setup is to what our brains do every day. We receive information from the world around us. Our brains transform it, change through experience, and produce some kind of output, whether that is a thought, a movement, a memory, or a change somewhere else in the body. Our brains have an extraordinary architecture, a body, senses, language, memories, and a lifetime of experience. But this experiment still felt like a stripped-down version of something all too familiar. And that is where the ethical discomfort or feeling kind of began for me. Is there a point where living neural tissue becomes too complex or too brain-like to treat as ordinary hardware, essentially a part of a computer? I don't know where that line is or whether this experiment has crossed it, but I do think it is a question we may have to deal with in the near future. And beyond the ethics, this paper left me with a more personal question. How computer-like are we? Sure, the organoid is not a tiny person, and I don't think this experiment means human beings are nothing more than computers. But it does make me wonder. Maybe the strange part is not that researchers made an organoid act like a part of a computer. Maybe it is that the experiment revealed in an extremely simplified form something about ourselves. As always, I can't wait to hear what you think about this, and until next time, stay curious and take care.